All right, so chapter 1 is going to focus on cell communication. And so cell-to-cell communication is really critical for both unicellular and multicellular organisms. This can occur through, especially with multicellular or multicellular organisms through adjacent cells that are connected via tight junctions or gap junctions. It can also happen through messengers. That is typically the way that cells are going to communicate with one another is via these signals. They're going to be called ligands. Those ligands are able to create a response that is going to be able to help the cell understand what it needs to do. There are definitely some mechanisms that exist across organisms, across species, across domains, because a lot of these signaling responses are going to be pretty universal to characteristics of living things. And so the fight or flight response, which is what you saw on the initial slide, is an example of how a signaling molecule can trigger that. And it's not going to be just triggering one particular process occurring in the cell, but it causes a range of activities to take place because with fight or flight, the goal is to survive. And so epinephrine is able to act as the primary signal molecule that gets things started. So external signals can be converted to responses within the cell using these signal transduction pathways, where we have a primary messenger, a ligand. It can bind to a protein on the membrane of a cell on its surface, or it can bind within the cell, depending on the type of ligand it is, that then can cause a variety of changes to occur for a specific response within the cell. Um, Again, it's thought that this type of signaling, because of the similarities in pathways, both with the prokaryotes as well as in eukaryotes, that this has been around for fairly early on in unicellular organisms, and then has just kind of evolved over time with our multicellular organisms and our eukaryotes. So one example, we talked about this briefly in class, are mating types for yeast cells, that they have mating factors that are secreted for the opposite type of yeast cell. You can have two mating types, A and alpha, and each of those signaling molecules that is secreted by that yeast cell are able to find receptors that are willing to accept them on the adjacent or the other type of mating cell, and that when those factors are bound to the receptors, there are changes that will take place. In class, I think we talked about it's a G protein receptor that kind of gets things going with this, that will lead to several changes occurring and induce the cells to basically fuse with one another. It's called a shmu. And then that will result in meiosis taking place because you will now have the chromosomes from both yeast cells joined together. So local signaling, again, can occur with the plasmodesmata, what we see with plant cells, gap junctions. There's all sorts of ways. We talked about these previously back when we talked about the cell structure, that cells are able to connect into the cytoplasm of adjacent cells. They can also use local regulators, which are molecules that are only going to be able to move over short distances. That can involve paracrine signaling, or they can actually signal themselves, autocrine signaling. And so there are, again, receptors present on the cell surface, and there's also signaling molecules that are present on the cell surface in the form of glycolipids and glycoproteins, which can help cells recognize one another. So in paracrine signaling, the molecules that are being secreted, the signal molecules, need to be near the cells that they want to target. While with autocrine, you will have molecules secreted by the cell that are able to bind to receptors on that actual cell. And these are known as local regulators. They just are able to move either to the same cell at different areas or to different nearby cells via the extracellular fluid. Synaptic signaling is what we tend to see with neurons. And so these are a little bit different because the shape of a neuron is not what you would typically expect to find with the cells that we've talked about previously. They still have a nucleus, but you can see there they have that really long axon, and then they have these areas at the end, which are called synapses. And depending on what type of electrical signal is moving through the neuron, it is able to secrete a ligand as a neurotransmitter that then can diffuse and bind to receptors on a nearby neuron or on a nearby cell and generate a response. So you can have neurons signal other neurons, or you can have neurons signal other nearby cells. These are known as synaptic signaling. And then long distance signaling is what we see with hormones. Both plants and animals are able to use this. It doesn't necessarily target a specific cell. These hormones are able to bind to a variety of types of cells as long as they have a receptor for it. And endocrine signaling is going to play key roles in maintaining homeostasis, being able to mediate responses to stimuli, and it's also going to assist with the regulation of growth and development of your cells. So with long distance signaling, these ligands, these signal molecules are going to be secreted from your endocrine cells. They move through the bloodstream, and then they can go into pretty much any body cell because these tend to be much smaller, nonpolar, so they are able to diffuse passively through the membrane and don't require any additional assistance. Depending on the type of signal molecule it is, some of them are able to move directly into the cells that they're targeting, but oftentimes if they are polar, they're just going to bind to the receptor on the external surface of the cell membrane. So Sutherland is responsible for describing how epinephrine is able to act on cells, and he is given credit for recognizing that when cells are receiving a signal, they basically go through a series of three phases. The first is the signal is received via the primary messenger, the reception, and then that signal is transmitted, is transduced, causing protein changes, other conformational changes, which can kind of set off cascades of signal transduction pathways. And then eventually, once all of the signal has been fully transmitted, so the first part would be if you were going to play telephone when you were little, would be the initial message that was provided to the first person. The transduction would be the number of people that that message moves through, and the response would be when the final person in the telephone call is told what they need to do. So here is a picture kind of looking at it when you have the signal molecule, the primary messenger, bind on the cell surface to its protein receptor, lock and key. You then would have relay molecules that are going to act as second messengers, and it doesn't matter whether you are the second, third, or fourth, or fifth messenger. If you are a non-protein, non-enzyme involved in this pathway, you would be given the name a second messenger. And then finally, when it's able to generate the response that the cell needs to perform, that's when you would have that response section take place. That could be causing a reaction to take place. That could be providing the mRNA that you need to make a gene or acting as the transcription factor to make that gene be transcribed so that it can be translated into a protein, or it could be leading to programmed cell death, apoptosis. So we're going to focus first on reception. So there are different receptor proteins that signal molecules can bind to. I use signal molecules and ligands interchangeably. The ligands are going to serve as your primary messenger, and they often are going to result in a change to that receptor protein. That is the initial transduction process that's going to occur. The majority of your signal receptors are going to be found on your membrane proteins, but the ones that are the ligands that are very small and nonpolar can move, diffuse through the cell membrane, and their receptor may be found within the cell, either in the cytoplasm or the nucleus, but somewhere in the cell. So these membrane-bound receptors are able to bind water-soluble signal molecules that would not be able to transverse the membrane as easily. And there are different classifications of membrane receptors, and we're going to talk about three. You can have a G protein coupled receptor, you can have tyrosine kinase receptors, and you can have ligand gated ion channel receptors. The GPCRs, the G protein coupled receptors, are the largest family of cell surface receptors that you're likely to encounter. These are used by those yeasts to make the shmu. They're also used with epinephrine and some other hormones. We also can see them used with neurotransmitters. But basically, if you look at this receptor, you see that it is made of 7 transmembrane passages, and you have a hydrophobic portion to it, and then you have hydrophilic portions. The hydrophilic portions are where you're going to find the binding site for your primary messenger, your signal molecule, and they're also going to be within the cell on the interface between the cell membrane and the cytoplasm, where you're going to find the binding site for the G protein to interact with this receptor. So how does this whole thing work? The G protein, when it's in its inactive form, is not going to allow anything to happen on its pathway. But when a signal molecule binds to that G protein coupled receptor, it's going to activate it, and that will allow the inactive G protein to bind to it. GTP, which is just like ATP, will come along and it'll transfer a phosphate to this G protein, and that will allow that G protein to then be activated. Once it is activated, it gets released from that receptor, and then the receptor becomes inactivated again, and the G protein that is active now goes for its target. In this case, it's going to activate an enzyme. When it binds to that enzyme, it's going to change its shape, it's going to change its activity, and that's going to set off either the cellular response, or that change in the enzyme may be able to result in a second messenger forming that would continue transducing the signal from your primary messenger. So the G protein can be reused, the G protein coupled receptor can be reused, the enzyme that was inactivated initially can be reused. So the signal molecule gets that G protein coupled receptor to change conformation such that it can bind the G protein that's inactive. Once the G protein is bound in its inactive form, GTP is able to activate it. The G protein that is activated gets released. The signal molecule gets released. The G protein coupled receptor goes back into its inactive form, and the activated G protein then moves along to activate another protein present in your cell. The receptor tyrosine kinases are going to consist of a pair of monomers. They're going to be forming a dimer that are able to generate enzymatic activity when the tyrosines are actually phosphorylated. And kinases are able to help with this because kinases transfer phosphate groups from ATP to whatever molecule that they are directing. And usually they'll be called some sort of blank kinase. So here they're being called tyrosine kinases. They are able to phosphorylate the tyrosine amino acids or the tyrosine residues. Because there are 5 of them that are phosphorylated, the receptor tyrosine kinases can be used to activate a lot of pathways or to allow a lot of pathways to be occurring simultaneously. So these are really helpful in being able to maintain characteristics of a cell that involve multiple pathways, such as cell growth or reproduction. And when we have issues with these tyrosine kinases, there has been evidence connecting mutations in these tyrosine kinases in cancer. So, kind of like with the G protein coupled receptor, we start with an inactive form of the receptor, and then a ligand comes along, that primary messenger, and you can see that part of the tyrosine kinase is found in the membrane, but that there's a good chunk of it that is found in the cytoplasm. Once that signal molecule binds to the ligand binding site, and it needs to bind on both monomers, then when it's bound, those inactive monomers become activated and they are able to join closer together and they form a dimer. At this point, the dimer is able to activate the tyrosine kinase regions for each of the monomers, and it causes each of those tyrosine residues to be able to be phosphorylated using ATP. And once all of those tyrosine residues are phosphorylated, there are relay proteins that can bind to those phosphorylated tyrosine residues. When they bind to them, it activates those particular relay proteins, and it may be a one step or multiple steps, but each of those relay proteins can generate its own cellular response, or they can work in concert together. The third major type of receptor we're going to talk about are the ligand-gated ion channel receptors. So basically, the protein itself, the receptor, is able to act as a gate, and it changes shape when a ligand binds to its receptor. These are pretty important with the nervous system. We'll get into this later on this year with the sodium potassium. We've seen that a little bit. You also see this with calcium. But basically, when a signal molecule binds to a ligand in this receptor, it allows, or it senses, is able to measure intuitively that there is a greater concentration of ions on one side of the membrane than the other. And so when that ligand binds, it's able to change the shape of this receptor so that the gate becomes open and those ions can move through this particular channel. And once the diffusion process has changed enough of the concentration difference between the extracellular matrix or the extracellular fluid and the cytoplasm, that ligand will be able to disassociate from this channel receptor and the gate will go back down. So here you have no ligand bound, the gate's closed, and then ions are able to move through when it's opened. When the ligand binds, it opens up, it takes the gate away, it changes the concentration pretty quickly, able to generate a response. And once the cell no longer needs more of that particular ion, the ligand is going to dissociate and cause that receptor to become inactivated and prevent additional ions from moving into the cell. I said that those were all membrane receptors. You can have the intracellular receptors for your small nonpolar molecules like your hormones. Again, these can be found in the cytoplasm or they can be found in the nucleus of your cells. And when these happen, they are able to act as transcription factors, and they can help to regulate specific genes being transcribed so that those proteins will be formed. So one example we have here is of testosterone. Testosterone has its receptor in the cytoplasm. The receptor and the hormone, when they are bound together, are able to move into the nucleus and bind to the DNA. So specific sections of the DNA are transcribed, and then that piece of mRNA can then be translated and generate a protein. So that's just going through those different pieces. And it doesn't have to be the whole receptor complex that comes in. It could just be that the receptor protein that is maybe it's found directly in the nucleus. And so you are able to bring the ligand in to create your piece of mRNA, or it could be that you just have, by activating the protein is able to move in. So we've talked about the obtaining your signal. Now we're going to talk about transduction. And again, you will have one single ligand can generate a pretty profound response. They do this relay process because it can cause the signal to be amplified. Using one molecule to make four, and then each of those molecules making four more can generate a pretty significant response in larger quantities. It would be an example of positive regulation. And so when you have these pathways that are multiple steps, they can either work simultaneously with one another, or they can work and result in different cellular responses that are related, or they can directly impact each other depending on what is being made as a result of that transduction process. So the molecules that are taking the information from the signal and relaying it from the receptor and turning it into response are primarily proteins. Your signal molecules are not going to be proteins. And so this is similar to the domino mentality that one domino kicks off another domino, et cetera, until you finally get all the dominoes down. And when you have this transduction process occurring, because you're transferring it between proteins, you typically will have a conformational change associated with your protein as it binds to that signal molecule. So ways that we can transmit is by phosphorylating proteins, which we do with kinases. They are able to transfer phosphates from ATP to the protein. And then we can inactivate our proteins that have become activated using protein phosphatases. Rather than adding on a phosphate group, these are able to remove phosphate groups. So these are able to keep proteins in either an active or an inactive form in your cell, depending on the cell's needs. So here you see a signal molecule, how it is binding to its receptor on the membrane, which sets off a relay molecule that activates protein kinase 1, which then can activate protein kinase 2, which then can activate protein kinase 3, and that can activate a protein that you need to generate a cellular response. And you can see the PP there, that's for the phosphatase, and that would be when these kinase proteins that have been activated need to be deactivated, and they would lose a phosphate to go back into their inactive form. So I talked about how primary messengers are going to be your chemicals. They are never going to be your proteins. The same thing applies with these second messengers. The second messengers are non-protein, water-soluble ions or molecules. They're small. They can move into a cell via diffusion. We often see second messengers when we are dealing with the G protein coupled receptors as well as the receptor tyrosine kinases. CAMP, cyclic AMP, or cyclic AMP and calcium ions are often common second messengers, but they are by no means the only second messengers. We are able to form CAMP from ATP. It's able to convert it by removing two of the phosphate groups and allowing the remaining phosphate group to form a cyclical arrangement with the sugar making up AMP. And then there are ways for us to turn CAMP back into just AMP. And we can do that using phosphodiesterase, where we add a water molecule and that allows the phosphate group to become separate from the ribose sugar. So there's lots of signal molecules that can trigger CAMP from forming. We see it in those G protein pathways. We also see it with protein kinases. CAMP is commonly used to activate protein kinase A, which then is able to, once activated, phosphorylate many other proteins. And we are able to impact metabolically on this CAMP by inhibiting adenylyl cyclase, which is what's going to be able to form CAMP in a G protein receptor system. So there's your G protein coupled receptor, something like epinephrine is joining, activates your G protein, and that activates, in this particular example, that adenylyl cyclase, which then can cause CAMP to be formed, and CAMP then can activate one of your kinases, leading to a cell response based on the cell's needs at the time. So calcium ions can also act as a second messenger. So you can bring calcium ions into your cell using ATP and having them move across their concentration gradient actively. The areas with greater concentrations of calcium are blue, the ones with lower concentrations are orange. And so by having the calcium ions move against their concentration gradient, we can increase the amount of them that are present in certain areas within a cell, and that can cause other cellular processes to occur. Some other second messengers we can run into are inositol triphosphate, IP3, and diacylglycerol, DAG. These two are part of a molecule, a glycolipid that is part of your cell membrane. So they are not a protein. But when these form, IP3 then can bind to adenylyl cyclase and help to activate, or sorry, it can bind to a ligand-gated channel, and it can allow ions to be able to flow in. And so that's what you're seeing in this particular example. You've got your G protein coupled receptor, your ligand, your first messenger has bound to it, activating your G protein, which in this case is activating the relay protein phospholipase C. Phospholipase C gets to cleave that phospholipid, the PIP2, that's part of your membrane, and causes it to make a DAG and an IP3, which also both can act as second messengers. IP3 can bind to the calcium channel that is gated by it, allowing those calcium ions to go against their concentration gradient, going from higher to lower, or sorry, going with their concentration gradient, I apologize. As it goes through there, those calcium ions can move in, and as they increase in concentration in the cytoplasm, they can activate different proteins, which can lead to different cellular responses. So we've talked a lot about the signaling and the transduction, but now we're going to talk about responding. What are the cells going to do once they've had this information brought to them? The response can have impacts on their cytoplasmic activities. It can also have impact on transcription, what is going to be formed from your DNA that then can be used to create proteins. So we can see responses occur both in your cytoplasm as well as your nucleus. And this is just one way your cell has to regulate cellular activities. It's able to turn enzymes on or off by turning genes on or off within the nucleus. It can also act as transcription factors. So there are pathways that regulate just your enzymes activity as opposed to just flat out synthesis. We tend to see those more in your cytoplasm. So here you have growth factor, which is bound to its receptor. It's going to move in and go through that phosphorylation cascade. Eventually, it's going to make it into the nucleus. Once it has gone through that phosphorylation cascade, it can activate transcription factors that are proteins present in the nucleus and cause certain pieces of DNA to undergo transcription. This signal can be amplified if you take a single epinephrine molecule back in reception and you put it through the various second messengers and the proteins that it's able to go through to activate those second messengers. And eventually you can have a response. You can have a single epinephrine molecule produce 100 million glucose 1-phosphate molecules, which then can move into respiration and generate lots of energy, which you would probably want to have available if you are in a fight or flight scenario. So aspects that can be fine-tuned with your response. You can fine-tune your amplification, its specificity, its efficiency, as well as its termination. So with these cascades, you are going to have positive regulation occurring, lots of amplification of signal, and the more steps you go through, the more amplification that's there. Depending on the types of proteins that are able to have these signal molecules bind to them, you can get different responses based on what receptors these signal molecules bind to, what activities they turn on or turn off in the cell. You can have relay molecules that are going to just result in one response. You can have relay molecules that result in two or more different responses. You could have a different ligand bind to a different membrane protein that creates a relay molecule that actually can either activate or inhibit another pathway. So that'd be crosstalk. And then you can have the same ligand bind to a different receptor, which would set off its own set of responses. The scaffolding proteins, you saw those a little bit with the tyrosine kinases. They basically are a large relay protein that other relay proteins can bind to provide its support, and so that you can have proteins that need to be made for a common pathway all together at once. So you make sure that they're all getting the signal simultaneously and they all know what the cells need. So sometimes these scaffolding proteins actually can serve as activators for the relay proteins. So here you've got your signal molecule, your receptor, there's your scaffolding protein, and you can see that you have three different protein kinases that have bound to that scaffolding protein. And so as the second messenger moves into the cell, all of those kinases then can send off either additional kinases or eventually your cellular response. You want to make sure that you can terminate the signal when you no longer need that particular pathway to be in place. And so inactivating mechanisms are going to be very important. When a concentration of a ligand falls, you're going to have less of the receptors that can bind that ligand busy and when are bound, and when they are no longer bound, they can become inactivated. So the final thing we're going to talk about is how cells know when they need to break themselves down. Apoptosis is programmed cell death or controlled cell suicide, and this is when cells have been targeted for destruction. We need to break down their nucleic acids, we need to break down their proteins, so that those macromolecules then can be used by other cells. And so they get packaged into vesicles. Why we want to have apoptosis available. It's able to keep enzymes that are only going to work under certain conditions from being able to come out and perhaps impacting on nearby cells. And you also can have cells that are dying off that are not going to be capable of reproducing themselves. And so you want to make sure that they don't negatively impact nearby cells. So in C. elegans, which is a soil worm, they were able to study this, and basically this is going to take place when the proteins that activate the controlled programmed cell death are going to be in a more active state than the ones that prevent programmed cell death. And so here you have a receptor for a specific signaling molecule that indicates the cell needs to be broken down. Within the cell, you have proteins that are inactive and active. In this case, CED9 is going to prevent CED4, which is going to be one of your proteins which is going to help to break down your cells. So just by having an active form of CED9, it prevents the CED4 and the CED3 from breaking down the rest of the cell. When a ligand, the death signaling ligand, binds to that given receptor, it will inactivate CED9. And because CED9 is no longer working to shut down the cells, it's going to activate those proteins that are going to be able to act as nucleases, breaking down your DNA, and proteases, which are able to break down a good portion of the rest of your cell. So caspases are your main proteases, the ones that cut up proteins. We talked about how you can have ligands that indicate that cells need to die. You can have damage to your DNA that's not going to be productive to copy it again. Or you could have protein misfolding that's going to be so substantial that it would not allow the cell to function properly. It's felt that this evolved early on in animal evolution, that it's really important for animals to be able to maintain and develop. And it's thought that this perhaps might be involved in some diseases, such as Parkinson's and Alzheimer's. And if apoptosis is not being regulated accurately, it could lead to some cancers. So here we see a frog, and you can see how you can't see the webbing between the individual toes, interdigital tissue, and then over time, those cells are recognized that they need not to be there anymore, and they go through programmed cell death. And now you eventually reach a point where you can see the individual webbing of the toes of this particular organism.